Thanks for joining us for another edition of Biotech and Life Sciences CEOs. Today, we're excited to have Raymond Peterson as our guest. Raymond is the founder and CEO of DNA Analytics. He brings over 20 years biotech experience to DNA's efforts to develop algorithms and genetic assays intended to help client companies improve and expedite genetic research and testing. Raymond's background includes time at the National Cancer Institute, where he led the development of nucleic acid modeling algorithms, which earned awards for research excellence, forming the basis for his first company, Celadon. In addition to his work at DNA Analytics, Raymond is active in supporting early stage ventures in the Washington, D.C. area, with insights from his experience in finance, business development, and operations. Raymond, I read that you recently raised some money for the American Lung Association by biking from Seattle to Atlantic City. First of all, what made you decide to bike like 3,000 miles? And
Well,
did you
really good
learn
question.
anything from the experience that might translate to entrepreneurship in the life sciences?
Yeah, could well be. Well, thank you for that great introduction. I really appreciate it. I'm really excited to be here as well. So why did I bike my across the country? Well, so in terms of the foundation with the Heart Institute, certainly we have had a heart disease in our family, and so it was very much on the top of my mind to do something like this. I actually did it a few years ago, and I was a bit younger then, and it seemed like this insurmountable goal that I just had to do, and that nobody was going to tell me I wasn't going to do it.
Well, so I guess when you start with an empty sheet of paper
Right.
and you design a company in the life sciences, why you need that sort of devil may care kind of
Yes.
attitude.
Right.
Man, you've used kind of the layout of the scientific problem behind the development of the COVID vaccines as a descriptor of the strategy behind the development of the DNA analytics product, which you call AccuMatch.
Yes.
Maybe you could kind of walk us through that journey as a way to a general description of what you're trying to do with AccuMatch. And first of all, can you tell us about some of the definitions? Like, for instance, what's a genetic probe and why is it important?
Very good. Okay, so that's an excellent question, and I will start there. So, a genetic probe in biotechnology is it's a short single strand of DNA or RNA that is used to query or to match a very specific gene sequence in our genome. So, probably as you know, our genome has 2,000 unique base pairs, 4,000 if you pair them up. And it's not so easy to get a gene probe that's unique to a gene. And the reason you would want it unique to the gene is if you're doing, say, diagnostics or gene therapy, you want to target that very, very specific gene and none others in the genome. There's only the four bases of chemical letters in the nucleic acids, the A, C, G, and T. A pairs with T, C pairs with G. And because you have such a limited alphabet there, when you have 2 billion opportunities, just from random chance, there's maybe going to be matches elsewhere in the genome. And so that's what our algorithms do, is we sort of scan the genome and we look at the biophysical properties of those genes and identify the best location in that gene to put that gene probe so that a diagnostic will have very clear signal for that gene. For example, say cystic fibrosis. You might want to know whether you have that gene or not, and so but you don't want it to be confused with another gene. And same thing with, say, gene editing or gene therapy. If there is a defective gene, say cystic fibrosis, and you want to change that and repair it, well, you only want to make that repair in that one location. You don't want to change other areas of the genome because that could lead to problems.
So I read today, as a matter of fact, I think it was in Endpoints, about some of the difficulties that the gene editing companies have had with AAV vectors
Yes.
and lipid nanoparticles and others that have caused, in some cases, the FDA to put a hold
Yes.
on what they're doing. Tell us about why a gene probe might be superior in terms of that targeting.
Yes. So there's a number of ways. One in particular, so those technologies that you described all use uh, gene probes, although they're not all known by that same name. So, for example, for gene editing, CRISPR is the acronym that people talk about, and so there it's called a guide probe in terms of like guiding to that gene. Um, so yes, there are a number of ways in which things go wrong. We've already touched on the one, which is if that guide probe matches another gene and makes an edit in another gene, that would presumably destroy the function of that and information in that other gene. But then also, some of these gene therapies have been potentially causing an immunological response in patients. That you put these, as you say, so right, so that AAV, that vector, that's a virus. We have to be able to get these guide strands into the cells. And so one delivery mechanism is to put these guide strands inside the shell of a virus, and then you infect the person with that sort of dormant virus, but that virus does attach to cells and then gets that genetic material, the guide strand, inside the cells. And then it does its business inside the cells. Well, that whole process could sometimes cause our immune system to recognize that as a foreign entity and either have a very strong immunological reaction, which sometimes they call that a storm, could cause like really severe fever and other side effects. But then also there has been some evidence that these putting these entities into the cell could somehow or sometimes um, causes the cellular machinery to act in such a way that the cell then becomes somewhat cancerous and divides uncontrollably. And so those are two main areas in which we have to be careful. And the way that a gene probe can improve on that is the best gene probe means that you would have to use a lower amount of it, sort of less drug in the body, and less drug then means that you have less opportunity for side effects. And then also we can design somewhat around of causing that immune response. And so
So,
were those the kinds of things that you were seeing? And maybe did you read about other
yes,
things this morning that I might
yes,
be able to comment on?
there was, well, I don't, um, it was Tessera, a company named Tessera.
Ah, yes,
Um, and they're, looking to move away from the uh, LNPs.
right, exactly. So interestingly, this past week was a very big social week in biotech because it was the JP Morgan Biotech Week in San Francisco, which I attended and gave a presentation for DNA Analytics. There's actually 4 or 5 conferences at the same time. None of them are large enough to accommodate the entire industry. And so my presentation was at the biotech showcase, and we talked to maybe something like 25 or 30 one-on-one partnering sessions with gene therapy companies. And many of them are interested in our technology because they do see how having better gene match algorithms in the design of their gene therapies will be able to give them a higher quality drug candidate in less time and for less cost.
Right. So that the match algorithms, which are, let's say, currently in use
Yes,
broadly, I guess date back to the late 90s to the early 2000s.
that's right.
And they have resulted as a result of, inaccuracies, um, giving bad designs, I guess, as many as 65% of the time.
Yes, that's right.
Um, what's been the effect in the clinic in terms of operations of those inaccuracies? Are they built into timelines in the clinic? Are they expected?
Sure. Well, so if by clinic you mean say the entire development process of
Right.
this drug, right? So there's several sort of stages to that. The first steps in research and discovery, of course, are to like sort of have your target of which gene it is that you want to modify, and then at the next step, that's where our algorithms come in. Is that where you need to design that drug to that gene? Once you design that drug, it's usually not a drug or a candidate. It's a little bit like high throughput screening with traditional pharmaceutical companies, where um, the development company will come up with maybe several thousand gene probe candidates or guide strand candidates, and then do laboratory testing of those candidates, which might take 6 months, a year or more, and require a very large infrastructure and maybe as much as several $1 million or more. And so that kind of limits the ability of this technology to those companies that have the resources to do that, maybe tens of millions of dollars it takes to screen. And at the end of that process, they winnow all of those candidates down to just several, and then those several candidates go into the clinic. And some of those will succeed, some will fail. And so what we're able to do with our algorithms is reduce that set that they test in the laboratory. So you spend a little bit more time in design, but you may only have to test 10 or 20 candidates in the laboratory. And then by the time you do get to your further experiments, putting them into animal models and humans, because you have better drug candidates, they then pass all of those criteria and gates as you go through with drug development, that yes, they look, operate well in cells, and they operate well in maybe mice, and then eventually into humans, and and to be safe and effective in humans.
So one thing I didn't understand as I went through your deck was the way that you described the improvement in accuracy, the way it was
Yes,
quantified. Can you give us kind of a layman's description of that?
sure. So when two nucleic acid strands come together, those A's match with T and G pairs with C, there's a strength of what we call hybridization. Hybridization means that they're coming together. And so that hybridization, the strength of that depends on the order of those DNA letters. Say A, G, C, T is going to have a different strength than T, T, G, G, T. And so that's what our algorithms measure. We did very specific and sort of controlled experiments in the laboratory, and generated the data for all possible matches over a short distance. And then with that, we were able to train our algorithms. So in the science, in the industry, they call it this match strength. So sequence dependent. It depends on the sequence. So the strength of an A matching a T depends on the letter that comes before it and the letter that comes after it. And so we measured all possible contexts like that. Others have done that as well. But what we did that's different is because it now is 20, 30 years later, we have improved genetic technology and more high throughput technology. So we were able to generate 20 times as much data as the prior algorithms. And then because we were able to generate so much data, we also were able to look at the statistical models that were used in prior studies, and we're able to improve those statistical models. So our algorithms are very similar statistically to prior algorithms, but have some improvements to them. And so when you add those two things together, the improvement in the statistics and the modeling of those nucleic acids, and then the vast amount of data, we're just able to become more accurate and precise than the prior algorithms.
So I think in the deck you implied at least a significant reduction in terms of time to market. Can you give us an idea in practical terms what that might be relative to current industry standards?
Sure. So it's anywhere from months to years of the improvement. So again, as I had mentioned earlier, you have this design, you have some number of candidates, you test and optimize those, or screen and optimize those in the laboratory. And so that process can take anywhere from 6 months to 2 years. Because the number of candidates are, say, something like 3,000 candidates, because we're able to come up with a much shorter or much smaller set to test in the laboratory, maybe 10 or 20 or 30, that ends up being maybe a matter of weeks to months versus many months to years. Yeah, so it is truly, transformatory and transformational. There is an analogy to our algorithms, which is in sort of the traditional pharmaceutical approach, which is called small molecules. And those are the drugs that were most of the drugs that are on the market. When we go get aspirin, that's a small molecule. Ibuprofen is a small molecule, and that's true of most of the drugs that are on the market. So these are very small chemicals that have either been by luck and found in nature, or designed in the laboratory to actually bind to, say, a protein in the body, and it's that binding to the protein in the body that makes the effect of the drug. So there's computational drug discovery that has been advancing over the past 30 and 40 years. And so for small molecule drug discovery, there's dozens of companies out there that are doing computational models to model in the computer the interaction of that small molecule and that protein, to see how they fit together. And then once you get a candidate protein, to tweak the chemical structure of that chemical to fit better into that protein and the sort of operational active site of that protein. And it's the same sort of value proposition benefit there, that by performing all of that computation, then in the laboratory, you may only have, say, 100 chemicals to try. And out of those, you get some candidate or lead or hit chemicals, as opposed to what previously was done, which is pharmaceutical companies were screening hundreds of thousands of chemicals. And so we're very similar to that. We use different molecules. Our molecules are the nucleic acid. So our drug is a short section of DNA or RNA, as opposed to that small chemical. But it's the same kind of conceptual thing, where we're able to apply these models to the heart of how that drug operates, which is the hybridization of that gene probe or guide probe to the gene, and where else it might hit elsewhere. And so one of the advantages, and why people have been so excited about nucleic acids, is that we more or less know our full set of genetic material. I mean, we've had the Genome Project, we've sequenced that. We know our messenger RNA. It's not perfect, we don't know everything, but we kind of have a complete picture of that, or almost complete picture of that. Versus for small molecules, we've got 100,000 proteins, and those proteins, every single one of them has a very complicated shape. And in order to do a comprehensive computational screen on the small molecule side, you would have to computationally see how that small molecule bound to the entire protein of all 100,000 proteins. And that's just computationally not feasible today. Whereas for us on the gene side, we are able to do more or less a comprehensive hybridization study, where we can see exactly how that putative drug or candidate drug is going to bind to its target gene, and also how it looks like it's going to bind elsewhere in the genome. So, and again, that's one of the excitements of the genetic medicine, is that we're able to have more of a complete picture kind of coming out of the gate, versus traditional drug discovery, where sure, you can show in a cell that that drug is highly effective. That's not why most drugs fail. It's not because of lack of effectiveness, but it's because of side effects. And it's those side effects that really get the drugs. And so with our algorithms, and also in this whole space, we're better able to predict those side effects and to design around them and to prevent those side effects from happening.
So your first company, Celadon,
Yes.
built software for the design of genetic diagnostic assays,
That's right.
which were ultimately licensed to companies like Illumina and Quigen and Millipore, that sort of thing.
Yes.
And it's been stated as having been the precursor to DNA analytics.
Yes.
How is DNA analytics kind of the next iteration of what you were doing at Celadon?
Right. Well, thank you for asking that. So at Celadon, the difference was that we did not have our own algorithms. We were using the industry standard algorithms
Okay.
that were, publicly available. These are generally scientists who had published articles, and we took the data from those articles. And it was during that work during Celadon that we became dissatisfied with those algorithms, and felt that there was a better way of doing things. And so that's why we went out and were able to secure funding for doing the sort of idiosyncratic laboratory work that was required to retrain and better train the algorithms. So what's different about DNA analytics is that we have very unique intellectual property. It's not just a matter of taking what was out in the literature and applying that in a good way. We actually are able to get better predictions because of our algorithms.
And what was the process, and how long did the process take toward developing that IP?
Right. So the process was somewhat long. It was a 5 to 6 year process. And the funding that we had was, well, there was a number of sources. The first source was the state of Maryland. This is something that some entrepreneurs might actually be very interested in, that the state of Maryland has a number of programs to help entrepreneurs, especially in high technology fields, that when I talk to people from other states, they're sort of astonished that Maryland has these programs. So one of the programs, and this is what started our algorithms, was something called the Maryland Industrial Partnership Program, or MIPS, and which is its acronym. And the way that it works is that the state of Maryland provided funding of $200,000 to a professor, Dr. Jason Kahn, at the University of Maryland College Park. The money went to his lab. Our company contributed some money to that. I think it was about a 10% match that we matched. And Dr. Kahn is the genius who was able to look at the existing algorithms and the data that was generated to train those algorithms and say, yes, we can do a better job. And so the work was initially started in his laboratory under that funding mechanism. We discovered with that, as we generated the data, that actually the existing algorithms were, in fact, could be improved, which was sort of doubted at the time. People thought that that the algorithms were as accurate and precise as they could be. And so we leveraged that information to then get additional funding from the National Cancer Institute under the Small Business Innovative Research Program. And so we had then won a $750,000 2-year award. And that's what really promoted and enabled us to develop the laboratory work and generate the data in the laboratory. As it turned out, what we had not anticipated was how difficult it would be to actually use that data statistically to generate our algorithms. So after those 2 years were done, and so now we're in year like 3 or 4, because actually 4, because we had the 2-year project with Maryland, and then it took another 2 years with the NCI. I took another 2 years to actually go through all of our data and to understand it as well as it needed to be understood, and to verify it and make sure that it was good data in the laboratory, sort of quality assurance, quality control, which we believe some of the other studies, which I'll get to in a moment, were not as good at that, or at least not clerically reporting their data. And then running through the algorithms and making sure that it was that everything sort of all tied together. This was a very clerically challenging project.
Yeah.
By clerical, I mean that we had many thousands of pieces of data It was a multi-step process, probably like an 8 or 12-step process to go from the signal and data that came off the instrument to an actual trained data set. And making sure that each step of the way we weren't making mistakes was just, just from a data handling perspective, very challenging. And then when we did train our algorithms, we said, well, and our statistical models and software pipeline to generate all this, we then took a step back and said, yeah, it looks good to us, but gee, let's look at the data that was published in the literature and take their raw laboratory data, which was published because these were academic papers, run it through the pipeline and see if we get the same answers they did, because we should. And we ran it through our pipeline, didn't get the same answers, which is then threw us into a loop for 6 or 9 months, because then we're like, well, heck, if we're using their raw data but not getting their end results, but
Right.
implementing their models that they published, like there must be something wrong with what we did. So we had to check every little step along the way. And as it turned out, we were able to show that some of the algorithms that had been published in the past were just super accurate. We were able to replicate their data to like the third decimal place. And then other articles in the literature, we weren't able to replicate their data, and we were off by or differed by 20 or 30%. We finally
Wow.
came to the conclusion with all of our own internal checks and quality assurances and looking at it from all these different angles, was that, they probably made a mistake clerically in reporting their results. And that's one of the reasons why the current algorithms aren't as precise or as accurate.
Wow. 20 or 30%. So what about the competition in terms of not only in terms of really the old way of doing things,
Right.
but is anyone else trying to do what you've already done?
Yes, so certainly. So there are some academics, some professors in laboratories who are doing basic discovery research, who work in this field. As it turns out, it's a very niche field. That's really surprising to me, considering how important these algorithms are to most of biotechnology. There's maybe 6 laboratories in the United States and maybe a dozen or two around the world that actually have generate the data to come up with new algorithms. So our competition are those academic laboratories, and then there are a number of companies. Ionis Pharmaceuticals is one that has done their own laboratory work and has developed their own algorithms based off of their own laboratory work. But the reason that it's perhaps a real niche mark or sort of area of science is that the statistics involved are sort of unique, and not many people are trained in that exact statistical models, which doesn't mean to say a general statistician couldn't look at it and pick it up, but the data that needs to be generated is driven by us thinking of a new model and then going into the lab and generating very specific data that is unique and cannot be used in genetic experiments. What other endeavors have done is, well, they've done the same thing we have done, but just on a much smaller scale, because it costs so much to do the laboratory work. They could only afford to do a smaller amount of data. But in sort of everyday activity, as people were designing assays and gene therapies to many, many different genes, they would get an idea of which ones worked and which ones didn't. And so they took those data sets and tried to figure out why some were working and some weren't. But because those designs were not made in a methodical way, it was just sort of, I guess, what's the analogy? You throw spaghetti against the wall and you hope something sticks. It's just like the quality of the data was just really haphazard. And it just didn't, it just doesn't give enough information to really derive something meaningful out of that.
So you've got an impressive list of clients. I mean, from diagnostic companies like Exact Sciences and Invitae, to gene therapy companies like Regenix and Spark, and vaccine companies like Merck and SmithKline. What have been the challenges in terms of acceptance? And what kind of company is more likely to look favorably
Right.
upon what you're doing?
Right. Well, thank you. So, yeah. So, some of the companies you mentioned are clients. Others are ones that we're doing pilot studies with, and others are ones that I sort of gave to you as a list of ones that are, sort of working in the space where we don't have a relationship with, but certainly would like to. So the resistance and the questions that they have is in part overcoming a, a bit of the disbelief that the current algorithms aren't as good as they could be because they've been in use for so long and so many people have relied on them and just not really thought much about whether they're accurate and precise. And then uh, most companies have something very unique about how they go about their process or their drug. And so no matter how many pilot prior studies that we have that show the efficacy of our algorithms, um, new customers often say, well, but we've got something a little different. How do we know it's going to work in our system? And so we point to both our laboratory data that we generated in training the algorithms and say, see, we've got this biophysical data that shows it's better. That's sort of like the with a car and putting it on miles per gallon and saying, well, we have more miles per gallon. Like, well, yeah, you did that on a laboratory test, but how do I know that when I'm driving around my town, I'm going to have better miles per gallon on this model? So we often have to go through a very short pilot project where we on a small scale charge a sort of a nominal professional fee. We make some designs and work with the customer, establish the relationship, and they try it in the laboratory. And generally, they get better results than they were getting before. And then they're willing to either license our software, which is what our primary business model is, or to do a larger project as a professional services agreement, more of like a partnering.
That was kind of going to be my next question is what the business model is. I know that Ionis is kind of they're looking for royalties, right?
Right.
You are more, I guess, of a SaaS kind of model, right? Or a
Yes,
consumption.
yes, a subscription model for
Yeah.
sure.
Is it a subscription or is it a consumption model? We pay when we use it.
Well, typically it's more a subscription model.
Okay.
Rather, so like an annual subscription rather than pay for it when we need it. And so the benefit of that is that then client has their own fixed cost. They know what that cost is, and they have as much use of that software as they need to. Now, the price of that subscription might depend on usage and how large the company is and how many projects that they're doing and that sort of thing. But the other reason that works is that there's a number of different ways that we could deliver the software. Certainly, it could be over the internet that on our servers or on Amazon servers, we make the algorithms available to customers. But some customers don't like that because they are very concerned about their intellectual property and they want to keep their intellectual property within their walls, including the internet walls. And so they would prefer us to deliver some software code to them that lives within their building and they can integrate it into their existing pipeline with one or two lines of code. And then it runs on their servers and they're not concerned about somebody stealing their intellectual property. Because really, for a gene editing company, gene therapy company, it's that guide strand, that gene probe, that exact sequence that ends up being the primary intellectual property that they have. That is the drug, just like the small chemical in traditional pharmaceutical companies.
But how do you avoid sort of that opposite concern that someone steals the code?
It's
So,
protected.
okay,
It's encrypted. Our
okay,
code is encrypted. And so it would, I mean, so no encryption algorithm is perfect, but they would have to, if somebody wanted to reverse engineer our code, it would take them, quite a specialty to actually decrypt the code.
interesting. Okay.
So
So,
that's how it's, yeah, the algorithms are encrypted, so they can't get at the actual code. And then what we supply to them is an example software code that's not encrypted, which they then integrate into their pipeline. But the actual library the code is encrypted, and it's only executables, and it's very hard to decompile.
okay, okay. So, Raymond, one of the things that I find interesting about yourself, really, aside from DNA analytics, is that you're active in providing strategic advice to other early stage companies in the DC
Yes,
area. If you had to boil it all down to one bit of advice, and I know that's hard to do,
yes.
but what would it be?
I would say that it's endurance. Stick to your guns. If you have something that's really useful, it's going to take usually a little bit to make people uh, realize the benefits of that, and it's going to take some time to develop that. And so you are likely to have hurdles in your way, as we all know. I mean, that happens every day in life, and you have to be able to figure out a way to overcome those hurdles. And in my case, the motivation for me was having sort of knowledge of the industry, and knowing how important these algorithms are and what a difference it could make to getting therapies to people, and seeing that there really wasn't anyone else pursuing this was just, it's like, this has got to be valuable. And just that sort of deep held belief that this is something that really is going to move the industry forward.
Right. I'm really surprised, or maybe this did happen, that, out at JP Morgan, you weren't surrounded by guys from CRISPR and Intellia and, Editas, especially Editas. They could really
Right,
use some help. Um, but
right,
but just, Raymond, we have to have this, and we have to have it now. So
right. Well, I agree. And so, I mean, so we were at that conference. I mean, we did have about 25 one-on-one meetings, not with the as some smaller companies, not the larger ones that you mentioned, but we so to get to larger companies like that, you generally have to have a little bit more of a track record. In a way, our this conference in just last week is our company's commercial coming out party, that we've developed the algorithms. We've gone purposefully slow. One of our customers, Switch Therapeutics, has now data from the laboratory that shows the efficacy of our algorithms. And that's kind of the next step that you need to attract the interest of other companies. Like, well, what's your data? Show us that your that it's not just speculative, that your value proposition actually is true. And we've been able to show that. And so now that we're able to show that, we're willing to go out and try to get meetings. We're not out so much getting meetings. We're waiting for this data from the early trials to come in, so that we could actually talk about something significant. And so when you said that, to get back to something you said before about, like, what's the one thing you would say to entrepreneurs? Well, one of the hardest things is to get those first customers, and to actually get that data that shows that there really is efficacy, because that's really what um, partners and customers want. They have to show them that there's actual value here, and that other people have realized that value. We've been really fortunate that we've had uh, um, scientists and clients who understood the basics and were able to look at our laboratory data and agree that that laboratory data looked well enough as to train the algorithms that they were willing to give it a shot. And they did, and it improved their process and it saved them time and money. So they were willing to license the software and continue with it.
Raymond, thank you so much for spending some time with us today. I really enjoyed getting to know you and getting to know more about DNA analytics, because I think that it can be transformative to an area which I personally believe is going to be transformational, not only to medicine, but to the economy.
Yes.
And we need that transformation.
We do. And
$31
yes,
trillion in
it's
debt.
healthcare, keeping people healthy, and a vibrant, robust economy. And all of those go towards, national competitiveness and well-being of both people in our country and around the world.
Right. And it really is just gathering in its infancy. It was interesting that you said, when I mentioned CRISPR and Intellia, you said that they were big companies. They're only, $4 billion companies.
Well, I
Those
get
are
it.
small
Yeah.
companies in the context of, for example, let's say DocuSign at
Yes.
30,
Right.
billion
Sure.
dollars. And I would submit that it's a nice product.
Yep.
Does enable you to avoid going to the lawyer's office and all that sort of thing. But
Right.
I don't anticipate it will change the world or the economy.
Right. So in the biotech industry, there are some bigger players like Genentech that are well known and sort of household names, perhaps. But a lot of biotech is surprisingly not exactly mom and pop businesses, but almost.
Right, right. Moderna almost failed, I think,
Yeah,
2 or 3 times.
right,
And
exactly.
prior to COVID was like a $500 million company.
Yes.
So um, it's a wonderful field. But anyway, to get back, just thank you very much for your time, Raymond. I really enjoyed talking with you and getting to know more about DNA analytics.
Well, thank you. I appreciate it. And thank you for the good questions. It was very nice to be able to explain, hopefully in relatively understandable terms, what it is that we do.
Thank you.
All right. Thank you.
Thanks for tuning in to the Life Sciences and Biotech Podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.